Hello and welcome to our video on cell signaling. So let's start with an overview. What is cell signaling? Well, cell signaling is the main process by which cells communicate. So there are three major stages of cell signaling. The stages are reception, transduction, and response. In this video we're going to go through these stages as well as attributes of these stages and explain the process chronologically. So the two main ways that cell signaling can occur are direct contact and travel by certain molecules to other cells. So first we have direct contact. So how can this happen? Well, first we can have cell-to-cell recognition, in which there are proteins, the membrane proteins sticking out of cells, they bind to each other, sort of like a lock and key fit, and they recognize each other, and then there's a sort of reaction that takes place. Certain molecules might travel between the cells and so on. This can also happen through cell junctions. So common cell junctions are plasmodesmata in plants or gap junctions in animals, and through these junctions various molecules can travel. And sometimes what's interesting is that viruses can manipulate these cell junctions in between the cells of a multicellular organism, and they can make those cell junctions larger. Like, for example, in plants, many viruses, what they do is they secrete these chemicals that increase the size of the plasmodesmata or plant cell junctions, and that allows the viruses to pass through even though they're relatively large. Just an interesting fact. So let's move. So next we have the fact that signaling molecules can travel between, into, and out of cells based on their chemical properties. So some properties, for example, let's take epinephrine for example, or as it's commonly known, adrenaline. So adrenaline is a water-soluble molecule. Now we know from our past videos that the cell membrane is really hydrophobic because all the hydrophobic tails of those phospholipids are facing inwards. So we have a very hydrophobic cell membrane. So epinephrine struggles to pass through the cell membrane because it is hydrophilic. So what happens is that epinephrine in general has a receptor on the outside of the cell, so that it does not have to go inside the cell because that would take way too long. On the other hand, you can have various hydrophobic molecules, which is a steroid, for example, which is hydrophobic, and those molecules tend to have receptors inside the cytoplasm on the nucleus, so that they cross the cell membrane into the cell. So obviously these properties are really important, and they've influenced the evolution of the receptors. So let's start with the types of signaling. So we have direct signaling, which occurs when two cells contact with each other, and a gap may form between them, allowing the passage of molecules. But we also have endocrine signaling, and that usually happens when we have hormones. So essentially these are molecules that are secreted by a cell into a circulatory fluid, usually blood, but it can also be other things, like, for example, in grasshoppers, it's hemolymph, which is a slightly different version of blood. And so this hormone, it recognizes a target cell based on proteins embedded in the plasma membrane or within the cell. So that's endocrine signaling. So paracrine signaling is far more local than endocrine signaling. So paracrine signaling is going to occur mainly when a cell secretes a molecule into a local cell, a cell that's very short distance. And for paracrine signaling, a lot of it is done through diffusion because the distance is very short. Then we have autocrine signaling, where the signaling molecule's target is the same cell, essentially the cell that secreted it. So there we have it. Alright, so let's talk about the first stage in the three stages of cell signaling. The first is reception, the second is transduction, and then we have a response. So let's start with the first stage, response. So the first step in the first stage of reception. So the first step in reception is the binding of the signaling molecule, and we like to call the signaling molecule a ligand. And so this ligand is going to bind to a receptor. Like I mentioned earlier, if you have a hydrophilic ligand, then the receptor will extend out of the plasma membrane, and a hydrophobic ligand will enter the cell and have a receptor within the cytoplasm. So there are three major types of membrane receptors, and those are the GPCRs, which stand for G protein coupled receptors, the receptor tyrosine kinase, and the ligand-gated ion channel. We'll start with the first one. So in GPCRs, ligands activate receptors, which then activate a G protein, and then that finally activates another molecule, which conveys the signal. And receptor tyrosine kinases, on the other hand, they form dimers. In other words, they come into pairs. So you have one receptor tyrosine kinase and another receptor tyrosine kinase. They come together, and then they break down ATP. They break down approximately 6 ATP, and then using those extra inorganic phosphates, what they do is they phosphorylate other proteins. Now, I remember a few videos back we talked about the various chemical groups, and if you remember that video, you'll remember that we talked about how phosphate is a very energetic type molecule. So if you add a phosphate group to another molecule, then that molecule is going to be very, very energetically active. And so that's why phosphorylation occurs, to make these molecules more energetically active, and so that the effect can be multiplied, and there can be a greater response for lesser input. So then we have ligand-gated ion channels. So these are channels that are permeable only to certain ions that open and close based on the binding of a ligand. So a good example of this would be in neurons. We have ligand-gated ion channels that are responsive to sodium and potassium and things like that. Alright, so let's move on to the second stage, which is transduction. So transduction, all it really is just when we convey the signal from the binding of the ligand to the receptor all the way to the final response, whatever that may be. So the way that we send the signal is through transformations in relay molecules. So that can be phosphorylation, which is the adding of phosphate groups, or it can be many other chemical transformations. Another thing I like to point out here, second messengers are these chemicals that speed up transduction substantially. And the reason that they do that is because they are really, really small. And so even though they may be hydrophilic, they can pass through the cell membrane really fast, and they can pass through the cytosol really fast. And so this dramatically speeds up transduction. So some good examples that you'll see a lot are CAMP, which stands for cyclic adenosine monophosphate. So it's sort of like ATP, but remove two phosphate groups. And you have adenylyl cyclase, which is also important to GPCRs, G protein coupled receptors. Finally, we have the response. So the response is essentially what happens because of the binding of the ligand to the receptor. What is the final output? So in some cases, the response is a modification to the genes, or to transcription factors, which essentially activate the genes. So what can happen is that when the ligand binds to the receptor, we have a vast chain of events, and at the end we have the activation of transcription factors. And you'll learn this later when we get into genetics, but transcription factors are really important to start translating the DNA and eventually resulting in RNA, which results in protein, and the protein is what actually executes the instructions of the genes. So that's why the transcription factors are really important. So, but there's another way that the response can influence the cell, and that is through modifications to proteins in the cytoplasm that already exist, that have already been synthesized. So there's another major phenomenon, that is signal amplification. So the reason that I have this exponential graph here, I want to illustrate the fact that it's sort of like a chain reaction. You have one event, one ligand binding to one receptor, and then that receptor activates various other relay molecules, and then each of those relay molecules activate many other relay molecules, and so on and so on. So it's sort of like an exponential function. So this can happen because the protein is going to be activating not one but many further molecules. That is why that when you look around you and a photon lands on your eye, which is what ultimately makes you see, that what you actually end up seeing is about 100,000 times as strong as the energy of that one photon that landed on your eye. That it occurs because of signal amplification, because the energy of that photon is being transferred from one signaling molecule to another signaling molecule, which is transferring to many other signaling molecules, and so on until we have a massive amplification. So I hope you enjoyed our video on cell signaling, where we covered the 3 major stages: cell signaling, reception, transduction, and response. I hope you'll join us next time.